Good morning everybody and welcome to our second webinar. We had one webinar already last week. This one will deal with the 7 truths of impurities and their reference standards. We will have a look today on a few of our 7 truths of the impurities and their reference standards. We'll deal with truth number 1 and the truth number 3 and 5 and 6. These truths will be part of this webinar today. The other 3 truths will be part of the webinar next week. So let's have a look onto the truth number 1 directly. ICH guidelines Q3A and Q3B need to be applied to generics as well. Before we come to that, a quick word to the ICH for those who are not familiar with it. That is probably nobody in that audience, but I would nonetheless explain it a little bit. It is the International Conference on Harmonization, and there you have sitting members from the regulation authorities and members from the industrial pharmaceutical associations from Japan, USA and Europe there, and discussing what is necessary to discuss on pharmaceutical quality, safety and efficacy. And the ICH published 3 guidelines so far that are dealing with impurities, and the most important ones are Q3A and Q3B. And there's Q3C also available, which is the impurity guideline for residue solvents, and that is already available in the 5th revision here. There's a 4th one under its way, the Q3D on heavy metal impurities, but we will concentrate today on Q3A and Q3B. Okay, so let's have a look on what is going on in Europe here before we come to the United States. When you look into the European Pharmacopoeia, there are 2 documents of importance there as regards ICH guideline Q3A and Q3B. And there are those 2 documents are the general monograph for substances for pharmaceutical use with the number 2034, and there's the general chapter for control of impurities and substances for pharmaceutical use. That's the chapter number 5.10. And what these 2 documents do is they link all the applicable monographs from the European Pharmacopoeia. These are called in the European Pharmacopoeia new monographs, and also we will see that later at the European Medical Agency. These are called new monographs, and the 2 documents above that you see above there in the slide link every applicable of those monographs to the ICH, Q3A and the thresholds there. And the thresholds, I put that table from QCA so here on the slide. The thresholds are given in that table here, and you can see that when you have a drug substance that is administered in the dossier, foreseen to be administered in the dossier with a dose less than 2 grams a day, then you need to report any impurity that is above the threshold of 0.05%. You need to identify and show the identification in your dossier for every impurity that is above the level of 0.10%, and you need to qualify any impurity that is above the qualification threshold of 0.15%. Qualifying an impurity means here to show that the impurity at the certain level, and this can well be above the qualification threshold, but at that certain level that you have it present in your drug substance, that it is not doing any harm to the patient, considering the duration of use and also considering the sickness status of that patient. So there are different rules applied when you have to treat a patient in a terminal status than just somebody who's having a cold, for example. There are also thresholds given for reporting identification and qualification for materials that are administered with more than 2 grams a day, and you can see those here. They are a little bit lower than the ones in the first row. When we leave European Pharmacopoeia and have a look then on the European Medical Agency, what they issued was a guideline on control of impurities in pharmacopoeial substances. And what the guideline is saying is that the not the inspectors, but those people that are looking on the marketing approval, that are responsible to grant the marketing approval for medicinal products, that they should grant that marketing approval only for those products where the monographs to which are referred to for the pharmacopoeial ingredients are already compliant with the two documents that I showed you in the slide before, 2034 and 5.10. What is also said there is, this is referring to the workers, to the staff of the EDQM, that the EDQM should not grant the certificates of suitabilities anymore when those requests from the applicants are still based on the old monographs that are not yet compliant with 2034 and 5.10. There are now most of these things. You see the guideline is from 2004, and most of the old monographs are now transferred to the new monographs, but still a lot of companies, especially those from China and India, still have problems to comply to the new monographs. The impurity limits changed a lot from the old monographs to the new ones. We will not see examples here today, but in the autumn webinars that we are planning, I will also give you a few examples there so that you can really see what changed from an old monograph to a new one. Okay, so that's what the case in Europe. What is the case in Europe? Let's have a look what is the case in the United States, and there we can look on the FDA. And FDA issued two guidances for industry on ANDAs, on abbreviated new drug applications, and that's the term at the FDA for generic substances. So FDA issued these two guidances here, impurities in drug substances, respective impurities in drug products. And there, what the FDA is saying there is that that they know that ICH Q3A and Q3B were developed for new drug applications in the beginning, but that the FDA is of the position that those principles from ICH that I mentioned there are also applicable to the abbreviated new drug applications to the generic products as well. And this is the quotation there, and important is the first sentence here, that the FDA believes that much of the content of the Q3A guidance applies to ANDAs as well. The FDA is saying in their guidance also something about how to set acceptance criteria for each and every impurity that is above the reporting threshold of ICH guideline. The first point of reference there in the guidances is the Pharmacopoeias, and the FDA is mentioning there the USP. And what the FDA is saying there is that the impurity, when you look on a certain impurity and you find it specified in the United States Pharmacopoeia, then you should keep the specification that is mentioned there. You should stay compliant with that specification there. It is not saying there in the FDA guidance that it would acknowledge pharmacopoeia specifications from other pharmacopoeias as well, but I can imagine that if you suggest such a specification to FDA, when you find it in the European Pharmacopoeia or in the British Pharmacopoeia, for example, and there's nothing in the USP there, that the FDA would be fine with such a specification as well. However, when the Pharmacopoeia specification from the USP, for example, cannot be kept, then what the FDA is saying is, look, you cannot keep the specification in the Pharmacopoeia, so please show me that the substance, the impurity in your drug substance, is not doing any harm to the patient for the duration of use. So please qualify this impurity for me with whatever data. And this data can be, I forgot to mention that on the slide with the table there for qualification, these data can come from literature. These data, however, can also come from studies, from toxicological studies that were performed on animals, for example, or on cell lines. Whatever is feasible there and suits your purpose is worth to be presented to the FDA in order to to let them consider an impurity as being qualified. What is to do when an impurity is not specified in the compendia? Then the FDA guidance is showing a decision tree, and this decision tree is based on the ICH design. And I show you quickly that decision tree on the next slides. I don't want to go into detail here during this webinar. We simply do not have time for that. But what I want to raise your attention on is these 2 points here, where you are obliged or requested to reduce the impurity either here to a safe level or here to not more than the qualification threshold. And reducing, you can of course do that physically, in that you either purify the substance, the drug substance, in order to get rid of that level of that certain impurity, or you can change the synthesis route for the API, or your drug substance supplier can do that in order to get a more advantageous impurity profile, or you can change the supplier for your drug substance in order to find one with a more advantageous profile. But these are already big wheels that you need to turn, and before you really turn these big wheels, it makes sense to really look specifically on each of the respective impurities, because what is normally done here until this point here is that between impurity and the corresponding API, the same analytical response was assumed. And this, you know, is for most correlations simply not true. So it makes sense to not assume same analytical response here between API and the impurity, but to look, when you really look on the levels of these impurities, and you're really obliged to do that, you really look then with a dedicated impurity reference standard on those impurities before you really take any further actions like purifying, changing the drug substance supplier, or changing the synthesis route, for example, or even go into the qualification studies here, where you can see that they can take, when you look at the general toxicity studies, that they can take 14 to 90 days, for example. Another month to evaluate the data, and then you have 4 months spent maybe on that impurity that you can have checked before and maybe come to a no action point here. Alright, that was what I wanted to tell you about ICH here, that it needs to be applied for generics as well. Let's have a quick look on the truth number 3 now, about Pharmacopoeia reference standards, and that still a lot of customers incorrectly assume that they can apply an assay of 100% when nothing is written on the label. Just a quick look here on the most common pitfalls with Pharmacopoeia materials. The materials from the Pharmacopoeias, and I mentioned here EP, chemical reference substances, and the USP reference standards, they are very often designed only for just a single purpose. Sometimes they are for 2 or 3 purposes and used in 2 or 3 monographs, but very, very often for just one single purpose. And what they are very necessarily not suitable for is that they are suitable for the non-pharmacopoeial purposes that you want to apply them maybe off the monograph there. And if you do that, the user is responsible for such an off-compendial use. The next slide shows the quotation from the USP and from the European Pharmacopoeia. On that, there are quotations in the USP General Chapter 11 and in the general text from the European Pharmacopoeia 5.12 that are stating that the assessment of the suitability for use in other applications than the monograph applications rests with the purchaser, and here in the European Pharmacopoeia that for any purposes other than that for which it has been established, the suitability for the CIS has to be fully demonstrated. And now we come to what we call here in our company the assay rumor that a lot of customers still really tell us. And what they are saying is, look, there's nothing written on the label, so I can assume the assay is 100% for my purposes. And that is really something which is not true. It is a misunderstood USP passage from the General Notices 5.80, and we need to read that very carefully. And it is saying, unless a reference standard label bears a specific potency or content, assume the reference standard is 100% pure in the official application. And the word official and application is, these two words are the most important ones here, because it means when the official application, for example, is just an identification test or another qualitative test like a system suitability test, any qualitative test for which it was designed and only designed and for which it is then 100% pure, it does not necessarily mean that it is then, of course, 100% pure for any quantitative purposes like an assay checking or checking on a level of an impurity outside the use of the Pharmacopoeia. So keep that in mind, please. The European Pharmacopoeia is not having such a statement, but they also have such an approach in place for some of their quantitatively used impurity reference standards. And the reason for that is that the Pharmacopoeial impurity standards, as well as the other reference standards for the Pharmacopoeias, are often designed only for qualitative use. And when they are designed only for qualitative use, then the assay is not known and can be far away from 100%. It can be that it is 95%, it can also be that it is just 90%. Whatever is suitable for the purpose in the monograph, with that purity, a standard is also released. So be careful with that. If you use it then, if you assume 100% when nothing is on the label for non-pharmacopoeial purposes, then that can result in an overestimation, sometimes in a serious overestimation of impurities. This can cause OOS results, especially if you change from one reference standard to another, maybe, but it can also lead you to unnecessary qualification studies with all the hassle that is behind that, like a toxicity studies of 3 months, for example. It is better to use a dedicated impurity reference standard than rather than the Pharmacopoeial materials. Apart from the Pharmacopoeia materials, with a detailed certificate of analysis, and we supply approximately 3,000 of such impurity reference standards in our pharmaceutical catalog for reference materials. And on the next slides, I would like to show you the certificate very quickly, though. I also showed certificates in the webinar last week. On the first page, you can see here some general information, the name of the substance, the formula, structural formula, the molecular formula, and the molecular weight, lot number, catalog number, how to store long-term wise, how it is appearing, that material, melting point if possible, and then also we give here the assay information that we calculate with an equation that you will see at the end of the certificate. Start then the real part of the certificate with the identity section. And our impurities are identified by help of HNMR and by the help of mass spectrometry, and also by the infrared spectrum. We provide that as well. Right, so we come then to the purity section. We check on the chromatographic purity normally with HPLC. You can see the conditions given here. That it was measured with a DAD detector at this special wavelength here, what was injected, the conditions that it is gradient profile here in that case, and then of course also we show the chromatogram itself, the area percentage report here, and the chromatographic purity as being 99.85% here. As you know, HPLC is not looking on water and on residue solvents, so we check on that as well. On the last page of the certificate you find this section here, water by Carl Fischer, and the residue solvents are estimated by HNMR. And then the final result is set up. You see it here, once again listed all the results, and then the assay rounded here to be 99.2%. And the equation that we used is also provided now on the newer certificates. You can see it here that from 100% we subtract the amount of water by Carl Fischer, we subtract the amount of residue solvents and multiply then with chromatographic purity. So that is what we provide with our impurity reference standards, a detailed certificate there. And the regulators accept our impurity standards. We can truly say that. We have a lot of customers that put our impurity standards into their dossier, and normally it is always for impurity testing, it is normally always accepted. So you see that here they are frequently used in dossiers and, as said, readily accepted by regulatory agencies worldwide. So that was already truth number 5. Let's come to the last one, truth number 7 for today. As said, in the truths 2, 4, and 6 we will deal with in the next webinar. So truth number 7, our impurity standards can be used universally and might be less expensive than you would think. Our impurity standards, they normally come in a unit size that is in most cases 100 milligrams, where the substances are more rare to procure or more difficult to synthesize. We go to a unit size of 50 milligrams, but we do not go below that so far. Because what most customers really want is they do not want smaller unit sizes. And when you use them for method validation and these things, you need 100 milligrams or at least 50 milligrams there. And the smaller unit size makes, of course, sometimes sense when you just want to do an identification, a quick one. But apart from that, 50 and 100 milligrams are unit sizes that our customers prefer over the smaller ones. They are coming with a detailed certificate. We have seen that in the slides before. And they can be used in almost any analytical application when it comes to testing on an impurity, to validate methods there. When we compare our materials to other commercial providers, and I don't want to go into detail here with this presentation, I did it a little bit more in the last presentation when I compared a reference standard to a so-called research material, but we do not go into detail here in this webinar here. On a price per milligram basis, other commercial providers are often more expensive with their materials, and it is always advisable to check the price and then the documentation that you get for that price beforehand, before you make your decision which impurity material you would purchase. And if you have a look on C of A's of other providers, then in most cases when you compare them to our certificates, there are less detailed there, and yeah, they have less information, and that can sometimes also, as I pointed out in the case study last week, that can also sometimes result in the rejection of an impurity reference standard when not enough data is provided on the certificate. And so we can really say that no supplier provides more details on a certificate of analysis when it comes to impurity reference standards. There are a few who provide the same amount of information, I would say, but then they are also normally more expensive than we are. Let's have a look when we compare our materials to those from the Pharmacopoeia commissions. When you compare them to the USP on a price per milligram basis, we are in most cases less expensive than the USP, and we of course provide a detailed certificate of analysis, and the USP is providing their USP certificate, which is not really C of A indeed. And as we have discussed already before, the USP materials are applied with restricted use, are to be applied with restricted use based on the monograph, for example. And when you compare our materials to the European Pharmacopoeia standards for impurities there, on a price per milligram basis, we have to admit that in most cases the European Pharmacopoeia is cheaper on their materials, but there you have no certificate at all provided with the materials, so not with the USP, where you at least get the USP certificate, but there is no certificate provided with the European Pharmacopoeia materials, only a leaflet sometimes advising about the use for how to use that material in the monographs. and you have the very restricted use for the materials from the European Pharmacopoeia, as most of their impurities are really set up for qualitative purposes like identification or like peak identification and system suitability testing. Right, so that was what I wanted to tell you here about the 7 truths of impurities, and the next 3 truths we will talk about in the next webinar next week. So a few closing remarks very quickly, because we have done that also in the webinar last week. LGC Standards is part of the LGC group, and our Science and Technology section at LGC is acting as the National Metrology Institute for Chemical and Biochemical Measurements in the United Kingdom, and to that regard we are comparable to the NIST, the National Institute for Standards and Technology in the United States. We manufacture pharmaceutical reference materials in our site here in Luckenwalde, nearby Berlin. For APIs and impurities, we have set up over 3,000 standards that are available from us and displayed in our catalog. We also set up metal impurity materials that are suitable for ICP and ICP applications here. Those we do not set up in Luckenwalde, but we manufacture them in our sites at VHG Labs in the town of Manchester, in the state New Hampshire in the United States. We constantly do new developments, and we are in the process of launching a range of primary reference standards for APIs in October 2013. So we have then materials available that can really be called primary standards for drug substances. We are ISO 34 and ISO 17025 accredited in Luckenwalde, where we set up those standards, and the primary standard range that I just mentioned to you is produced under the ISO Guide 34 and the ISO guidance 17025. We have contract services available. We do custom synthesis for reference standards. We do custom analysis for materials. We characterize customers' materials if they want us to, so they provide us with the material. And we do also customized packaging and storage. And on the next slide you can see that very quickly. So here you can see all the steps that are necessary really to go through in order to provide a reference standard for an impurity or for a drug substance, doesn't matter. But these are crucial steps that needs to be done. And what we also do, and in the next webinar next week I show you a few case studies there, what we do for customers who either just book certain parts of these sections here. So somebody is just choosing characterization and certification and takes the rest he takes care of by himself. Others are asking over the whole range, and I would like to show you a few case studies there. And also, I hope this is interesting for you, also prices with which you can calculate then for such services. I say thank you, and I'm waiting for your questions.